Okay. Hello everyone. Welcome to free crash course for competitive exams held by VIRAT Hindustan Sangham in association with Manifest IAS Bengaluru. This program is inspired by Dr. SUBRAMANIAN SWAMY and is convened by RAVISHANKAR Sir, State Education Convener for VHS Karnataka branch. I am DHANUSH KUMAR, Chief Mentor of the program. So we're done with the 5 major topics, dealing with the 6th topic, that is general science. So it includes both science and the application part, that is technology. Both of these things I'm going to discuss. So what is the source for this? See, for science and technology, there is no specific source as such. For your state and central level public service examination, there is no specific source. So they say NCERTs can be referred, but there is lot of excess information which is not required from the perspective of competitive exams. So the best thing is pick up any science and tech material of any good institute, compilation of any good institute or any good author. That is more than sufficient. You can base it on the reviews of the book either in Flipkart or Amazon. So that would be sufficient. No need to break your head on this, right? So yesterday class I initiated the topic with the biotechnology. So we were discussing the structure of a cell, the difference between the plants and an animal cell, all those things we discussed. So today application part I'm going to talk about is the genetics part. So genetics. So what is genetics? It's a branch of biology. It forms a branch of biology. It deals with the genes, right? So humans. So if there is a difference between me and you, it is the genetic difference, right? So between me and another animal, it is species difference. Genes also differ as well as species also differ. Whereas when it is between two humans, the genes are going to differ substantially. Primarily the DNA part. DNA is going to differ, right? So genes get transferred from one generation to another generation. That is called as genetic transfer of genetic information from the parents to children. That is their offsprings, right? So this deals with the hereditary. So the genes are hereditary. They flow from grandparents to parents, from parents to grandchildren, right? It is going to flow across hereditarily, right? So genetics basically deals with the hereditary transformation of genes, transfer of genes from one generation to another generation. So the basic unit of genetics is a gene. Gene is the basic unit of genetics. So if you have to study your genetics part, you have to begin with the study of gene. So what is a gene? Gene is a set of nucleotides. Nucleotides, it is found in the DNA. Genes are found in the DNA, right? So I'm going to show you the picture of the, see, this is an organism. Human, this is a human or any other animal, right? So humans are made up of tissues are made up of cells, right? So in cells, each cell has a nucleus. So in the nucleus there is chromosome. Chromosome, I told you, they are thread-like structures. Within the chromosome there will be sections of genes, right? Each chromosome is one long DNA. So each chromosome is made up of one DNA, entire DNA, and genes are functional regions of this DNA. So this is the entire DNA. In that, the specific parts are called gene. So gene is the next part of the chromosome, right? This entire part is DNA. The specific part is gene. So DNA is double helix. Double helix andre DNA is spiral. It is in the form of, okay. So you know the gene diagram, right? It is in the form of spiral. So the DNA will be somewhat like this, double helix, and there will be base pairs. This is the basic diagram of a DNA, right? So these genes, what they do, they encode proteins. The function of the DNA is to, gene is to protein. See, all our function, metabolism is based on protein synthesis. Who produces the protein? So even though you eat the food, it is getting converted to energy and the energy is supplied to the cell. In the cell, the genes are producing the proteins, right? The essential proteins. So if specific part of a DNA doesn't produce protein, right, they are not called genes. So genes means they definitely produce proteins, right? So these genes actually vary in size, shape and everything. There are approximately the bases, the gene bases, that is these staircase like structure, these horizontal structures, there will be approximately 2 million of them. 2 million are the number of the thing. So gene expression. So next is gene expression. What is gene expression? How the gene gets expressed from parents to their offspring is called as gene expression. Hege? So it might be in the form of a physical structure or behavior or any specific disease or anything. So that is the gene expression, gene getting expressed itself, right? So the genetic code of a parent is going to get expressed in the offspring. That is the expression of genes, right? So the genes, how do they express? They get converted into proteins. So they produce the proteins are transferred from one generation to another generation. That is genetic expression. But please remember, genetic expression is how the genes get expressed from one generation to next generation. That is the expression of genes, right? Okay. So then the gene expression includes two processes, basically two forms, two main stages. So one is gene transcription. What do you mean by transcription? So the production of RNA, messenger RNA. So you know DNA, one more is RNA, ribonucleic acid. If they produce the messenger RNA, that is the transcription. The gene getting transcribed from DNA, it gets converted to mRNA. mRNA is going to be used for genetic expression, right? So that is transcription form. One more is translation form. Translation, this mRNA, the first stage is transcription, DNA getting converted to mRNA, right? Messenger RNA. Second stage is translation. The mRNA is going to get converted or it is going to produce the proteins necessary for the synthesis of necessary for metabolism. That is the translation. That is the second process. So now we are going to talk about genetic engineering. What do you mean by genetic engineering? So application of engineering, that is technology to alter the genes, addition of genes, remove of gene, transforming gene from one form to another, that is genetic engineering, right? So next the concept is gene mutation. So gene is going to get mutated. So especially when these ultraviolet rays are there, so they say if they fall, if the ozone layer is not there, these ultraviolet rays are going to fall on the skin and the DNA is going to get mutated. Mutated means, so it is going to get converted into some other form. That is gene mutation, right? So if there is a change in the DNA sequence, that is called as DNA mutation or genetic mutation. So this is a bit because of the mistake made in the DNA copying. So copying, the DNA gets copied itself. So copying, if there is some mistake because of any external factor, environmental stress agirbodu, or it might be the ultraviolet rays, like that, any external reason, if the copying mechanism, DNA getting copied itself, if there is any problem in that, then it is called as gene mutation, right? So DNA, if there is a change, that is called as mutation, right? Okay. So this mutation generally it doesn't get carried from one generation to other generation. If it get carried from the parent generation to children generation, it is called as germline mutation. Germline mutation. Germline means the offspring from the parent to the children. That is germline. So genome transformation. So only if there is mutation, that is germline mutation. The concept is very, very important. Then genetically modified organisms. So whichever the organisms in which the genes have been changed, or if there is any transformation, that is genetically modified organism. So under this there is transgenic organism and living modified organism. So genetically modified organism andru onde, transgenic organism andru onde, living modified organism andru onde. So all these mean the same thing, right? So this genetic modified organism, it can be either the plants or animals or anything. Anything can be a genetically modified organism. Then DNA. DNA means, what do you mean by DNA? Deoxyribonucleic acid. It is a chemical. So it is a chemical name for molecule. It is a molecule name which carries genetic instruction from one generation to another generation, right? So it is a chemical name of that molecule. It is basically a molecule. So it carries the genetic instruction. So there are two strands. Two strands andre, like, so these are the strands. This is one strand, this is second strand. So this is called as double helix structure. There is double helix structure in the DNA, right? So this double helix, this base is made up of sugar. The base is made up of sugar, deoxyribose and phosphate. This is made up of deoxyribose and phosphate. These two bases, these two structures. So it is better I write it in different colors. This base is made up of sugar, deoxyribose and phosphate. It is made up of deoxyribose and phosphate, and it has got four bases. Here there will be, in this staircase like structure, there will be four bases. What are the four bases? Adenine, cytosine, guanine and thymine. These are the four bases. Adenine, cytosine, guanine and thymine. They are the four bases. So they are held together by bonds. So there will be, these two are held together by bonds called as adenine bonds or thymine bonds. So these four bases are very, very important. They are going to ask adenine, cytosine, guanine and thymine. These four things, these stairs are made up of four bases. So that is adenine, cytosine, guanine and thymine, right? So this, what does the DNA include? It includes instruction. It includes instructions for assembling proteins and RNA molecules. It has instructions encoded in it for protein synthesis and also RNA, producing the RNA molecules, right? So this twisting, this DNA is a twisted structure. Actually, here also there will be lot of twisting. Here it is there, but the picture is very small. So this twisting is because of water present in the cell. Why the DNA gets twisted? And it is because of the water, hydrophobic and hydrophilic interaction. Andre water jote the DNA is going to interact and this structure will be formed. That is, twisting structure of the DNA will be formed, right? So who discovered the DNA structure? So four people actually discovered the DNA structure. So the names, two names are very important. One is James Watson and Francis Crick. James Watson and Francis Crick, these are the two people who discovered the structure of DNA. So along with that, Rosalind Franklin, Maurice Wilkins, they are also involved in the discovery of structure of DNA. Next, I'm going to talk about RNA, ribonucleic acid. So RNA, so in DNA there will be double base, whereas in RNA, what will be there? There will be a single base. So there will be single base and there will be single structure. This is the diagram of the RNA, right? So why it is also a polymer, it is also a biopolymer. It is a living polymer. So what does it do? Andre, it is also expresses the genetic information, genes. It is going to use for decoding, encoding the genes, right? So there are multiple types of RNA. What are the types? mRNA, that is messenger RNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. These are the three types of RNA. So all these things have their own special functions. Messenger RNA has its own function, transfer RNA has its own function, and ribosomal RNA also has its own function, right? So this RNA is also going to produce the proteins. It is going to produce amino acids. Amino acids are nothing but proteins. So this amino acids in turn produces the protein. RNA produces amino acids. Amino acids only what will be there? Nitrogen. So that is in turn going to produce the proteins, right? So this RNA, first there will be DNA. This DNA is going to get converted to RNA, and this RNA is in turn going to, that is mRNA, is going to produce the protein, right? So this is the basic mechanism. So what are the differences between DNA and RNA? What is the difference? So one is, name is the difference. It is deoxyribonucleic acid, that is ribonucleic acid, right? So this DNA, there is long-term storage of memory. So genetic information will be stored for very long time in the DNA, whereas here in RNA it is stored for a temporary time. That is for making the proteins, until the proteins are synthesized, till then only the RNA, till the production of proteins only the information will be saved. After that the genetic information won't be saved, right? So then here in the bases, so the base is made up of, in DNA it is made up of what? Sugar as well as phosphate. So here in RNA also it is made up of sugar and phosphate. What are the base pairs? Base pairs in DNA, I told you, adenine, guanine, cysteine and thymine. Whereas the base pair in RNA, there is a change. Adenine is there, guanine is there, cytosine is there. Whereas thymine is not there. In the RNA, there is no concept of thymine. Instead of thymine, there is uracil bases are there. So what are these adenine, thymine? These are all chemicals, various chemicals, okay? So instead of the thymine, there will be uracil bases present in the RNA. So whereas DNA is what? Self-replicating, it replicates by itself. Whereas RNA is synthesized by the DNA, it is not self-replicating. So DNA in the produce agat RNA, it is not self-replicating. So, and I told you the difference in base pairs. So here it's the same thing. The 4 same bases will be there except thymine. Instead of thymine in RNA, there will be uracil acids are present. So DNA is susceptible to ultraviolet rays damage, whereas RNA is not susceptible to ultraviolet rays, or it is resistant to ultraviolet damage. So this is the concept of RNA, the difference between DNA and RNA, right? So evolution of biotechnology. Now I am going to talk about how biotechnology, that is technology applied to the biology, how did it evolve over time, the evolution of biotechnology. So in the evolution there were multiple methods in the evolution of biotechnology. What are the multiple methods? So first, the basic biotechnology was microbial fermentation. Fermentation of beer, fermentation of wine, fermentation of milk, all these products approximately. So this kind of technology, fermentation, is there since 10,000 years, right? So since 10,000 years, all these things are there. So whereas in modern biotechnology, so these are the basic types of biotechnology. Whereas in modern biotechnology, you have genetics, biochemistry, molecular biology, and all these. So there is higher level of technology in modern biotechnology, right? Okay. So how did the biotechnology evolve? So in the last century, in from 1900 till 2000, how did biotechnology evolve andre? So first, the term biotechnology was, who gave this term biotechnology? In the year 1919, the name was given by a Hungarian agronomist called Karl Erke. He is the person who gives the name biotechnology, not very important. Then in 1928, what happens? The penicillin is discovered. Penicillin, antibiotic use. It is a first antibiotic. So who discovers penicillin? Alexander Fleming. Fleming discovers the penicillin. Then in 1943, DNA was discovered. DNA was discovered by, DNA was there, but someone said that DNA is the carrier of genes. DNA carries the genes. Anta. In 1943, they discover, right? So then in 1953, they discover the double helix structure of the DNA. This structure they discover in 1953. So then in 1969, an enzyme was synthesized artificially. Artificial synthesis of enzyme takes place in the year 1969. So then in 1983, first genetically modified plant, genetic engineering plant was produced in the year 1983. Then in 1997, first cloning of mammal takes place. First cloned sheep. The sheep name is Dolly. So this sheep was cloned in the year 1997. In 1998, there is a human genome map. Andre human genes na they are going to map it in the year 1998. So human genome map takes place in the year 1998. Then in the year 2010, what happens? First synthetic cell, andre artificial cell, animal cell is produced in the year 2010. And in the year 2013, a bionic eye was produced, an artificial eye was produced, bionic eye, right? So then in the year 2020, biotechnology is used to fight COVID, right? Okay, so this is the evolution of biotechnology since the 20th century, right? So what are the types of biotechnology? How many types of biotechnology are there? See, there are multiple types of 7 types of biotechnology, each demarcated in its own color. Various colors are given for it. So first is red biotechnology. Red biotechnology means health sector. The biotechnology associated with health is called as red biotechnology. Andre production of vaccines, production of various medicines, So all this stuff. Then green biotechnology. Green biotechnology associated with agriculture. Agriculturally pest control and various other stuff. So pest control agirbodu to improve the yield, high yield varieties, and to reduce the insects, right? So there, and also to reduce the intake of water as well as fertilizers. So all these technology can be employed in agriculture, that is green biotechnology. Then white biotechnology, what is it related to? Industries. So biotechnology applied to industries is white biotechnology, that is manufacturing sector. That is industrially what they do, biofuels, treating of the exhaust, recycling and all those things, treating the polluted waste, that is white biotechnology. Then yellow biotechnology deals with food production of the food. Then blue biotechnology, that is related to aquatic life, that is marine and ocean life, right? So aquaculture and all those things. Also I told you that seagrass and algae are used for fertilizers. So that is also, that can also be considered as green or blue biotechnology, right? Then gray biotechnology means recycling of the waste, recycling of polluted products is gray biotechnology. Then gold biotechnology is bioinformatics, that is DNA fingerprinting, DNA studies. So information level use of genetics is gold biotechnology. Okay, so these are the 7 colors and the meaning of them, 7 types of biotechnology. Then what is the application of biotechnology? Where all biotechnology is applied. One is, the most important sector is medicine. So starting from your COVID vaccines to hepatitis vaccine as well as insulin, everything. So hormone vaccine, so insulin, production of insulin, everything is based on biotechnology. Then industry, it is used in industry as well. So in industry, you have various types of concrete, right? Self-healing concrete and also production of clothes, production of footwear, right? So in industry, various types of products are manufactured. So they are based on biotechnology, right? So then food, genetically modified organism, genetically modified crops, high yield variety crops, that is with respect to food biotechnology. Then environment, what is the use of biotechnology? Bioremediation process, andre cleaning the environment, that is biotechnology related to environment. Restore the contaminated ecosystem, right? Restoring the ecosystem which has been contaminated, that is the use in environmental use of biotechnology. So how is the biotechnology sector faring in India? Bangalore is called as what? BT. What is BT? IT, BT, biotech, right? So India is the 3rd largest destination for biotechnology research in the world. So 3% of the global biotechnology industry, various industries, biotechnology research industries, medicine, pharmaceuticals, all these things, 3% of them are there in India, right? So biotechnology industry by the year 2025, it is proposed to hit $150 billion. So it has got huge potential of growth, right? So what are the sectors? Especially the pharmaceutical sector, the generic medicine, that is your, what is that? Janaushadhi, generic medicine, there biotechnology is used, and also biosimilar medicine. Biosimilar medicine, andre, it is the same same medicine produced in foreign countries, which is highly costly. But same biosimilar medicine, chemical medicine. So in medicines, what will be there? Chemical composition. Instead of the chemical, there is some other biological component, natural component, that is biosimilar medicine. So there also biotechnology is used. So when it comes to biotechnology, which is the regulating agency in India, biotechnology, it is Department of Biotechnology, DBT, Department of Biotechnology. When was it started? In the year 1986. 1986, it is part of Ministry of Science and Technology. Then apart from that, there is one more organization called VIRAT, Biotechnology Industry Research Assistance Council. VIRAT. So this Biotechnology Industry Research Assistance Council, very important body. This is also functioning. It is an autonomous body. It is a not for profit body and also autonomous body. This is under the Department of Biotechnology. So it focuses on research and innovation, right? And also funding the biotechnology products. What? VIRAT. Then what are the government initiatives to promote biotechnology in India? What has the Indian government done? So there is a National Biotechnology Development Strategy 2015 to 2020. National Biotechnology Development Strategy 2015 to 20, MBDS, right? So what is the aim of this biotechnology development strategy? Andre, to make India a world leader in biotechnology, right? India, that is the goal, right? So it aims to, what does it do? This strategy prakara, they aim to promote more and more funding, and they aim to promote more and more research, education, right? Bring in more and more biotechnology products. All these things are the aim of this biotechnology development strategy. So what are the features? Features of this strategy. One is promote biotechnology human resource. Andre, how many of the people, experts do we have in biotechnology? Very less, right? So increase the human resource in biotechnology. That is the first goal. Then research. Research is the second thing. Third thing is use biotechnology for inclusive development. Andre, use it for developing of all sections of society, right? Then commercialize the biotechnology. Andre, use biotechnology for making profit. It is also there, right? And use biotechnology for welfare of society, health needs, medicine needs, all those things. Okay, then foster cooperation in biotechnology. Andre, cooperate with other countries to increase the biotechnology. So these various things. And also he says use biotechnology in the research of human genome. That is the set of all genes, all DNA is called as genome. One genome is the set of all genes, right? So you study the genome using the biotechnology, produce vaccines, and also use stem cell research. So stem cells, what is a stem cell? This is one kind of cell which can take the form of any type of cell. So this, if there is a damage in some disease is there is a concept called stem cell therapy. So they are going to insert the stem cell and do therapy so that the damaged organ gets rejuvenated. I am going to discuss stem cell in some time, right? So all these things are the strategy, all these things are the goal of the National Biotechnology Development Strategy, hangover of environment. I am talking biodiversity. Okay. Then what is the next initiative? National Biopharma Mission. What is biopharma? Pharma and medicine. Biopharma means use of biotechnology in the medicine. That is National Biopharma Mission, right? So it is a joint mission between industry and academia. Andre, the research institutions and the industry, both of them have together started this National Biopharma Mission. So under this there is a Innovate in India program, I3 program. Innovate in India program anta there is a program, right? So what is this program andre? Encourage indigenous development, local development of biotechnology, applied medicines, that is Innovate in India, right? So who is implementing the biopharma mission, national biopharma mission? It is VIRAT. I told you VIRAT is the organization. I told you what is the full form of VIRAT. So this mission, national biopharma mission, was started in the year 2017. 2017 is the start of the year, the start of this national biopharma mission. Next, there is one more program called biotechnology program for Northeast region. So the biotechnology in the Northeast region is biotechnology program in the Northeast region. So Northeast region, it is actually a biodiversity hotspot, alva. Indo-Myanmar border, it is a biodiversity hotspot. So they say out of the total biotechnology budget, 10% of the budget should be spent for Northeast region, developing biotechnology in the Northeast region. So there is lot of biotechnological resources. Use that resources to create more products. That is the aim of this Northeast biotechnology mission. So under this there are one authority set up, that is Northeast Region Biotechnology Management Cell. There is a cell for biotechnology management, and also this is about biotechnology, national biopharma mission, and also development of biotechnology in the Northeast region, right? So where all does the biotechnology can be implemented in India? Where all can it be used? Can it be used to solve the problems of poverty, hunger, right? So various other things can it be, so the biotechnology can be used to solve the problems. So first is nutritional security. Nutritional security means food security. So they say 15% of Indians are undernourished. Who say it? Food and Agricultural Organization says 15% of Indians are undernourished. They don't have proper nourishment, and 38% of children are stunted. What do you mean by stunting? Low height for the age. If the person, so there are three things: stunting, wasting, underweight, and fourth one is anemia. So stunting andre low height for age. So ಈ ಹಳ್ಳಿಗಳ ಹುಡುಗರು ಚಿಕ್ಕ 10ನೇ ಕ್ಲಾಸ್ ಓದ್ತಾ ಇರ್ತಾನೆ ಅಂತ ಇಷ್ಟೇ ಉದ್ದ ಇರ್ತಾರೆ. So it is stunting. There is no nutrition. Underweight andre there is no proper weight for the particular age. Wasting is low weight for the height. For that particular height, if a person is having low weight, that is wasting. Anemia andre lack of red blood cells, RBC. So anemia is witnessed in women mostly. So they say more than 50% Indian women are anemic. So when the women are anemic, what happens? The children will also be born with nutritional deficiency. So that is the thing. So what are the programs we have for elevating the nutrition? We have Poshan Abhiyan, National Nutrition Mission. Also we have ICDS, Integrated Child Development Scheme. That is their ICDS. And we have Pradhan Mantri Matrutva Vandana Yojana, Pradhan Mantri Shishu Janani Suraksha Yojana, Shishu Suraksha Yojana. All these yojanas we have, right, to increase the nutrition levels. So even under your this thing, National Food Security Mission, so they gave nutritious food for mothers under National Food Security Act, NFSA. National Food Security Act, which year? 2013. So just before the Congress lost the power in 2014, they brought this act, National Food Security Act, under which approximately 70% of Indians get free food grains, right? Their nutrition is also given for women, right? So 38% of children are stunted and 21% of them are wasted. You know what is stunted and what is wasted? I told you, direct question in exams. Multiple times they are asking What is stunting, what is wasting, I've told you already, right? So how can biotechnology can be used to solve this problem andre? See, this one strategy is food fortification. Food fortification andre increase the nutritional quantity of food. So in the PDS system, public delivery system, what they are giving just food grains. Instead of food grains, you give fortified food grains. If instead of giving wheat, give fortified wheat. Fortified wheat andre add iron and all other nutrition to the iron, zinc, magnesium, whatever is needed, add it to the food grains and give it to the under PDS. So that is food fortification. So for that addition of nutrients, what is needed? Biotechnology is needed, right? So then where else biotechnology can be used? To reduce environmental degradation, that is bioremediation. Bioremediation andre no. So using of microorganisms, microorganisms, if you release in the polluted environment, they are going to convert the waste into water and carbon dioxide. So that entire region will get remediated. So there this biotechnology can be used, right? And also this biotechnology can be used to reduce the air pollution, right? So this particulate matter PM 2.5, they can be captured using biotechnology, right? So various methods are there. You need not, so just know where all biotechnology can be used. Then tackling health problems. So I told you stem cell therapy, gene therapy, there biotechnology can be used. Then even biotechnology can be used. See, there was this dengue causing bacteria. What is that mosquito? Aedes aegypti, na? So Oxford University, they designed one strategy. What they did was, for this Aedes aegypti, they devised a male Aedes aegypti mosquito. So these male mosquitoes, they used to mate with the female mosquito and make them infertile. Infertile andre they won't be able to reproduce. Though that kind of mosquitoes they produced in the Oxford University and they released it. But later on this project was shelved and I don't know. Maybe because of some other reason. So in that kind of things, the biotechnology can be used, and also biotechnology can be used for preventing diabetes, cancer and all those things, right? So it can be used to regenerate the pancreas. Andre, you give certain types of medicine to pancreas, there biotechnology can be used. So in agriculture, next is agriculture. Where does biotechnology come in handy for agriculture? One is drought tolerant species. Drought tolerant species of agriculture, biotechnology is used, and also pesticides, biopesticides. Biotechnology pesticides can be manufactured. Then reducing harvest losses, reducing weeds. So all these areas biotechnology can be used. Then energy needs, where biotechnology can be used on the energy, biofuels, biodiesel, right? Biodiesel, biopetrol, bioethanol, there biotechnology can be used. So what are the challenges for biotechnology sector in India? What challenges is the sector facing? One is the budget. In the budget, there is very less allocation for biotechnology, right? So science itself, the entire science gets just 0.67% of the budget. But if in Western countries and other countries, the budget is 3 to 4%, which is very high compared to India. Then next is IPR issues, intellectual property rights issues. Andre, if there is any discovery in the biotechnology, it will be patented. So they patent and they sell the product at very high cost. So it is not available for the poor people, right? So that patenting is the second issue for development of biotechnology. Then third issue is, and also third issue is compulsory license. I told you what is compulsory license in economy. So if a foreign country doesn't allow India to manufacture, if a foreign country doesn't allow the reduction of price of a medicine, or if it doesn't allow India to manufacture any generic medicine, India can give compulsory license. It can override the patent. If a particular medicine is having patent over it, that patent can be overridden by the government. That is called as compulsory licensing. When can they override the patent? When there is a public health emergency, they can override the patent. So these compulsory licenses are also a threat to biotechnology, because see, you discover something, but government gives it to everybody. So who will discover? So nobody will spend money on discovering. So that is one thing, right? Okay, so then there is no commercialization of biotechnology. Biotechnology is limited just to the universities. That is one issue. There is no awareness about biotechnology. So what can be done to improve the biotechnology? One is awareness. Second is, we can bring in a biotechnology regulatory authority. There can be a separate, instead of VIRAT, there can be some other authority which will help rejuvenate the biotechnology sector, right? So we can bring in a biotechnology bill as well, and the entire science and technology sector should get at least 2% of GDP should be given to science and technology, right? And also human resource development should be encouraged in the biotechnology sector. Not only Bangalore, many places should become biotechnology hubs. Then only biotechnology is going to grow. So now we spoke about the science part of biotechnology. So now we are talking about application part, application of biotechnology. Where all can the biotechnology can be applied? So one is DNA profiling. What do you mean by DNA profiling? Determining the exact pattern of DNA in a person, exact pattern of base pairs. So that is DNA profiling. So the DNA of each person is different, right? So determining the exact pattern of DNA is the DNA profiling. So see, if you take the entire DNA, most of the DNA are safe. But there are specific regions where there is high changes, high diversity is there. Those regions are called as polymorphic region. Polymorphic region of the DNA are having high diversity. So if me and you are differing in our genes, so we are differing in the polymorphic region of the DNA, right? So where can the DNA profiling can be used? So one is crime detection, rape cases and all these areas, DNA profiling can be used. Then to determine the dead body. So if there is a dead body, who is the person belonging to dead person, there it can be used. Then what else? In science and technology it can be used, and also determining the parent. So if a child is disowned by their parents, sent outside by their parents, the person can go claim it. So many incidents have happened. Later on they take the DNA test to establish that he is my father or he is my mother, right? So that can be done under the DNA profiling. So with respect to DNA, use of DNA, recently government brought in a bill. So this bill gives the entire procedure of using of DNA, how DNA can be used within the geographical boundary of India. So this bill is called as DNA Technology Use and Application Regulation Bill 2019. DNA Technology Use and Application Regulation Bill 2019. So this is also called as DNA Profiling Bill. This was passed by Lok Sabha, later on referred to Joint Parliamentary Committee. I have told you what is Joint Parliamentary Committee. It is a committee of both Lok Sabha and Rajya Sabha. That is Joint Parliamentary Committee, right? So this, where is this DNA? What is the purpose of this DNA bill? Andre, DNA profiling bill andre, to establish the identity of a person. So who person's identity, who are these parents, where does he come from, Has he involved in any crime? All these things can be identified. So what is the objective of this bill? To trace the missing person, to determine the offenders, and also to determine the deceased bodies. So all these are the uses of this bill or goals of this bill, right? So even in organ transplant it can be used. Organ transplant, if a heart is given from one person to another person, so to determine whether the heart is going to fit into that person, whether the kidney is going to fit into that person, their DNA profiling can be used, right? So what are the provisions of this bill, DNA profiling bill? One, it will establish a DNA regulatory board. All India DNA regulatory board will be established, right? So who will be the chairman? Chairman of this DNA regulatory board, it is Secretary to Department of Biotechnology, DBT Secretary. DBT is a Direct Benefit Transfer. Department of Biotechnology Secretary is the chairman of this DNA regulatory board, and also Director General of CBI. Other members are CBI DG, NIA DG, all these are ex officio members, right? So this DNA regulatory bill, what does it give? It is going to give a unified database. So database of all the DNAs that are collected within the geographical boundary of India, it is going to maintain. This is called as National Data Bank. National Data Bank of DNAs. Not only National Data Bank, it is also going to maintain regional DNA data banks. Regional data banks also it is going to maintain, right? So this every data bank, they have to maintain specific indices, not very important. So what indices andre? Crime scene index, suspect under trial index andre. So which are the cases where which are the cases involved in crime? Which are the DNAs involved in crime? Which are the cases involved with dead bodies? Which are the cases involved in determine of organ transplant? Like that, they have to maintain separate indices in the DNA bank, right? So under this, there will be DNA clinics. So these DNA clinics will be getting license or registered by the DNA regulatory board. DNA regulatory body will be the regulatory authority, right? So if the police have to take your DNA, they have to take your written consent. You have to give it in writing. But if you are convicted for any case which entails more than 7 years of imprisonment, 7 ವರ್ಷಕ್ಕಿಂತ ಜೈಲ ಹೆಚ್ಚಿಗೆ ಜೈಲು or death sentence idre, then you need not take your permission, right? That is the condition. So, and also once the person is acquitted andre, if he is resolved, if he is sent, what is acquittal? If he is declared innocent, then his DNA profile should be removed from the data bank, right? Based on a written request. So what is the significance of this bill? I told you what are the uses of this bill anta heladnalla. All these are the significance of this DNA bill, right? So what are the dangers of DNA bill? Government has brought in a DNA bill. What are the dangers? See, in science and technology, more than the exact scientific part, they are going to ask the application part. Application and exact science, both they will ask. So I'm going to cover both of those things. So DNA, RNA is pure science part, whereas DNA regulatory bill is application part, right? So what are the concerns regarding this bill? Privacy, that is our personal information can be misused. So if we have any disease or if we have anything which need not be disclosed, it might be disclosed to public. That is one thing. And also second thing is data might be leaked. So in the DNA data bank, there will be DNA data stored. That data might be get leaked because of hacking. Then third thing is, because of now only there is lot of communalism. So they might use this DNA to segregate people on religion. So Hindu DNA, Muslim DNA and all those things, they might do that kind of segregation. Then one more concern is this DNA profiling is very high costly mechanism. Then there is the next concern is there is no good infrastructure. Infrastructure is lacking for DNA. That is DNA data banks. DNA banks set madake there is no infrastructure. Then possibility of misuse. This our DNA data can be misused by the government. And also it might lead to violation of human rights. If they have information on our DNA, what is it? It is a violation of human right. If they don't take our permission and take our DNA data, it is a violation of our human rights. So what can be done to correct this? Consent. That is, government has to take our permission to collect our DNA. Then there should be data protection law. So if they collect the DNA, it should be protected. Only by a court order the DNA should be disclosed, or else it should not be disclosed, right? So there is this bill is pending, personal data protection bill anta ide. So this bill was recommended by Chris Gopal Krishna committee. So this bill, public data protection bill, it is, personal data protection bill is B.N. Sri Krishna committee. Public data protection bill, Chris Gopal Krishnan, right? So this is about the DNA bill bought by the government of India. So bought in the year 2019. Next I am going to discuss gene editing of the genes. What do you mean by gene editing? So in the DNA, if you remove specific parts, if you cut specific parts and install some other part, that is gene editing. What is the need of gene editing? Why gene editing should be done? See, most of the diseases, they have genetic origin, right? So especially the genetic disorders, they have genetic origin, right? So in order to cure from the disease, you can alter the DNA. Gene editing can be used, right? So this gene editing was used earlier also, but earlier what used to happen? They used to insert the gene anywhere they want, but now at specific regions gene can be inserted. So what is the technology? The technology name is CRISPR-Cas9. This is the technology for gene editing. CRISPR-Cas9. So recently the scientists got Nobel Prize for discovering this CRISPR-Cas9 gene. So who are the persons who got Nobel Prize? The Nobel Prize got in 2020 for discovering this gene. Who are they? They are called genetic scissors. And you know normal scissors, right? This CRISPR-Cas9 is a genetic scissor. To alter the genes, this scissor is used. So who are the persons who discovered this? Emmanuel Charpentier and Jennifer Doudna. EMMANUELLCHARPENTIER. So this person and Jennifer, JENNIFER DOUDNA. These two people discover the genetic scissors, right? So what do they do in this genetic scissor? So this is the diagram of genetic scissor. So see what happens. This is a DNA, target DNA. They are going to remove a specific part and insert another part, another section of DNA. So see, the genetic scissor is Cas9. This is the genetic scissor which is cutting the DNA, right? Okay. So how do they do it? So what is the full form of CRISPR-Cas9? So the full form is clustered, regularly interspersed, short palindromic repeats. Very big name. Clustered, regularly interspersed, short palindromic repeats, and Cas9 andre CRISPR associated protein 9. That is the full form. So how does this CRISPR-Cas9 genetic scissor work? What is the working mechanism, right? So what do they do? First, see, they want to bring in a change in a DNA. How do they bring in the change? By causing mutation in the DNA, by causing a mutation. How do they cause the mutation? How mutation? Mutation andre change. So first, there will be a guide RNA. Guide RNA andre it acts like a GPS. So this guide RNA is going to show this CAS9 for this CAS9, this guide RNA first what does it do? It goes and get attached to that specific section of the DNA which needs to be cut. First, this guide RNA goes and gets attached to the specific section of the DNA. Later on, this CAS9 scissor goes and cuts that specific section. So, what is the role of guide RNA? It is going to show the place where the cutting should take place. So, it is going to guide where the CAS9 is going to go and cut the DNA. That is the function of guide RNA, right? So they are called, the CAS9 is called as what? CRISPR CAS9 is called as molecular scissor. Molecular scissor. It is going to cut both the strands of DNA. That is the upper base and lower base. Both these things would be cut by this CAS9, right? So I told you the function of guide RNA. Guide ribonucleic acid is going to show the place where the DNA needs to be cut, right? So what they do, they remove the unwanted DNA part. Unwanted DNA na remove madi, they install the required DNA. This will do what? This is going to make the person healthy. So it is going to cure the person of disease, and that will be the benefit of this genetic editing, gene editing, right? So once a new DNA part is insert, what will happen? So there will be, it is kind of a operation, right? It is kind of a operation. So the cell is going to recognize that there is some internal change. So internal change admele, it is going to repair it. Once it tries to repair it, the new part will become part of the entire DNA. The new part will get adjusted to the entire DNA. This is the process of gene editing. What are the uses or advantages of genome editing? What are the advantages? One is treating human disease. Treating of diseases is the first advantage, right? So many genetic disorders are there. So sickle cell anemia, thalassemia. So all these genetic disorders can be treated with the gene editing. That is the first function. Second, what else can be done? So majorly it is used to treat the diseases. Gene editing majorly. So and also to prevent the disease, genetic disorder from transferring from the parent to the children. That is also function of this gene editing. So next, what are the developments in biotechnology? So these are till now what I told, these are the basic developments. What are the advanced developments in genetic, that is biotechnology? One is pharmacogenomics. This is the first area. Pharmacogenomics. So what is pharmacogenomics? See, if you take a medicine, the same medicine will not impact everyone similarly. So it is going to, the impact of the medicine depends on the individual person, right? It varies from person to person. So pharmacogenomics is going to determine how a particular person's body is going to react to the medicine. That is the pharmacogenomics field, right? So So, if a person is having any genetic disorder, how the body is going to respond? If the person is having different kind of DNA, how the body is going to respond to the medicine? All these things is the field of study for pharmacogenomics. So, pharmacogenomics, what are the uses? One is, see, once they determine how the medicine reacts with a particular body, what can they do? They can produce tailor-made medicines. Tailor-made medicines andre for a specific disease, for a specific body, specific person, particular medicine will be designed. Andre, if a person is having fever, they won't give paracetamol. They will give a medicine specifically for that person, made for that person. Just like tailor stitching the clothes for you, not buying ready-made clothes. That specific medicine can be done. Then what else? New drugs can be discovered based on pharmacogenomics. New drugs can be discovered, right? So, chromosomal diseases, all those diseases can be cured. Then, better vaccines can be found out because of pharmacogenomics, all these things. So, that is the first area, new area. Second area is vaccine production. So, when it comes to vaccine production, there are many types of vaccines. So these types of vaccines are especially important when we are reeling under the COVID. So the first type of vaccine is subunit vaccine. What is a subunit vaccine? See, in this subunit vaccine, so there will be pathogen. Pathogen andre virus, bacteria, fungi, all those are called as pathogens, which are alien to the body. So that pathogenally specific part will cause the disease. So that specific part will be taken and that part will be purified and put in the vaccine. That is subunit vaccine. Subunit vaccine for which diseases? Hepatitis B disease, HPV disease, all these things. Then the next type of vaccine is bivalent vaccine. Bivalent, and there will be two antigens. Two different antigens will be there. Two of them will be put into the vaccine. Antigen, what do you mean by antigen? See, if there is a pathogen which enters the body will produce what? Immune system, immunity will be produced. So it will produce certain kind of hormones or certain kind of chemicals. Those chemicals are called as antigens. So antigens are specific chemicals to attack the foreign alien body. That is antigen. So in bivalent vaccine, there will be two different antigens will be present in one vaccine. That is bivalent vaccine. So bivalent vaccine used where? Dengue fever, BCG, adenovirus, all these things. Dengue fever, BCG and adenovirus, the bivalent vaccine is used. Then DNA vaccine. DNA vaccine, what is the use? DNA vaccine, what will be done? See, DNA vaccine, the DNA, I told you how DNA will be converted into protein. DNA will be converted into messenger RNA and messenger RNA is going to produce the protein. So that is DNA vaccine. DNA vaccine is going to produce the proteins required to combat the disease. That is DNA vaccine. So the one disease called melanoma disease, that can be treated with DNA vaccine. So there are multiple types of diseases. Vaccines. Conjugate vaccine. What do you mean by conjugate vaccine? See, conjugate vaccine only, it is just like bivalent vaccine, but the difference is they are going to combine two antigens. One is weak antigen, another is strong antigen. These two antigens will be combined and given to the patient. So one of the antigen, so weak antigen is the actual antigen. The strong antigen acts as a vector. Vector andreno. It acts as a carrier. So strong antigen will keep the strong antigen here. It is going to keep the weak antigen here, and this strong antigen acts as a transporting agent. It transports the weak antigen within the body. That is conjugate vaccine, right? Then, So DNA recombinant vaccine. Recombinant vaccine. What do you mean by recombinant? They are going to ask what are the types of vaccines. Recombinant vaccine. What do you mean by recombinant vaccine? So this recombinant vaccine uses DNA technique only, similar to DNA vaccine. DNA technology. So it is in this recombinant vaccine, they are going to specifically design certain bacteria or certain yeast which can combat the disease. Specific bacteria or yeast is manufactured using DNA technique, and this bacteria or yeast is put into a vector. Vector andre transporting agent, carrier, and they inject it in the human body. That is recombinant vaccine. Then RNA vaccine. What is RNA vaccine? That is mRNA, messenger RNA is put inside a protein or some carrier vector, and that is given as an injection. That is RNA vaccine, right? So then toxoid vaccine. What do you mean by toxoid vaccine? So this toxoid vaccine only, they are going to insert the toxins. See, all these vaccines, they are using what? They are using the bacteria, I mean the toxins only. Andre, it is using the bodies which are not needed. So all the organisms which are not needed by our body, they are using. Why? See, when they insert this virus or bacteria into our body artificially, then only our immune system will recognize it and fight it. Once it fights it, what happens? It is going to remember the particular virus or bacteria. So if the virus or bacteria is attacked in future, then it is going to launch an attack immediately. So the best example is your polio vaccine. Polio vaccine andre no, they are giving polio virus to you. So polio virus natural ag bandi, you will get affected with polio. Instead of that, while you are healthy only, they are giving you polio virus. So so why don't you get affected by polio if they are giving polio virus? Because this polio virus would be recognized by the immune system and it is going to launch an attack and kill the polio virus. So avaga our body will remember the polio virus. In future, if the polio attacks, it is going to kill the polio virus immediately. So polio virus, there are 3 types of vaccines. One is oral polio vaccine. Andre bayal aktaralla, that is one thing. Then a live attenuated vaccine. Live attenuated vaccine andre, there will be live polio virus in the vaccine, that is live attenuated vaccine. In the polio virus, one more type of vaccine is there, inactivated vaccine. Inactivated vaccine andre, the polio virus will be there but it will be dead or it will be sedated, it will be sleeping. So that virus will be injected, that is inactivated vaccine. These are the 3 types of vaccines for polio. So I was talking about toxoid vaccine. Toxoid vaccine andre, see whenever a foreign body enters our body, what happens? It is going to release some toxins. Those toxins will be recognized by the immune system. So that kind of toxins is present in the toxoid vaccine. Then one more type of vaccine is nucleic acid vaccine. Nucleic acid vaccine, where will be the nucleic acid present? It is present in the nucleus. So that nucleic acid of the bacteria or virus or the fungus is going to be inserted in the carrier and injected. That is nucleic acid vaccine. Okay. So then what else is the use of this biotechnology? What is the new uses of biotechnology? So genome sequencing, if there is any microbes, so virus, this COVID virus, they did what? Genome sequencing. Andre, what is the exact arrangement of the genes? Genome sequencing and enilla. So you know the genes, right, inside the chromosomes. So these genes, how they are arranged in the DNA. That is the genome sequencing. So genome sequencing alli this biotechnology will be used. Then molecular cloning, andre inserting some other new gene, there biotechnology can be used. Then recombinant DNA technologies, andre that is recombinant DNA technologies and the production of vaccines. In the production of vaccines, this can be used. Then what else? What else? That is vaccine. Vaccine aitu. Then even the lab animals, to produce the lab animals, different types of lab animals, this biotechnology will be used. So all these are the uses of biotechnology, right? So then so this is the vaccines from animals or vaccines from viruses. So there are also vaccines from plants, vaccines made from the plants, right? So plant-based vaccine technology. So what they do, they are going to take a specific gene from a plant and insert it in the vaccine and give it to animals. That is plant-based vaccine, right? So what they do, what these plant-based vaccines do, they are going to generate some specific characters. And plantally there will be some genes which can cure some diseases. So those things will be put in the vaccine and given to animals. That is the plant-based vaccine, right? So see, gene from a human pathogen is inserted that infects the plants. So so they have made it complex here. What they do andre, okay, gene from a human. So actually one is from the plant gene will be inserted humans. Other thing is human gene is inserted into the plants. So if there is a diseased cell, diseased gene is there, that will be put inside the plants, and that plant will develop what? It is going to develop immunity for that particular disease. So when you eat that plant, what will happen? You are automatically going to get immunity against that particular disease. That is plant-based vaccines. So next I am going to talk about vaccines for COVID-19. So two vaccines got emergency use authorization by, who gave the emergency authorization? Central Drugs and Standards Committee, CDSCO. CDSCO gave the emergency use authorization for vaccines, right? So which are the vaccines available? You know, right? Covaxin and Covishield. So they are going to ask about Covaxin and Covishield. So Covaxin, it is the first indigenous vaccine in India. First India made vaccine. Who made this vaccine? Bharat Biotech and National Institute of Virology. They two combined and made this vaccine. Bharat Biotech and National Institute of Virology, as well as, where is National Institute of Virology? Hyderabad. Or Hyderabad or Pune? Let me check. I am also confused. National Institute of Virology. It is in Bangalore also. It is in Pune. Headquarters is in Pune, right? So National Institute of Virology and ICMR, Indian Council for Medical Research. So these three bodies made this vaccine. So it is an inactivated vaccine. Inactivated vaccine andre, the live coronavirus is killed and put into the vaccine, right? So why do you kill the virus while putting it into the vaccine? Because once you kill the virus, it will take away the replication capacity. It is not going to replicate. That is the use of killing the virus, right? So this inactivated vaccine is onde alla. There were many vaccines, hepatitis A, pneumonia, all these things, right? So what happens? see, this entire COVID, coronavirus is put inside the vaccine and given as injection. So for the entire COVID, the body will develop a immunity. Body is going to develop immunity, right? So this Covaxin has much better protection. Andre, it is going to entice much better antigen. Antigen andre, you know, immunity. So it is going to entice much better antigen, and it is effective against mutant strain also. Mutants. So mutants andre no. So virus is replicating in different forms. First there was what? First there was some varieties, then came this Delta variety, then Omicron, now some other variety. So the virus is mutating, changing its form. So that is DNA mutating. Or so which is the carrier of coronavirus? It is RNA. RNA is a genetic material, not DNA, right? So what is the concern of Covaxin? It has not yet received phase 3 trial approval. Phase 3 trial approval data, na, they have not yet made it public. Then the next virus, next vaccine is Covishield. It is manufactured by Oxford, AstraZeneca, and what else? Serum Institute of India. They manufactured it, right? So this is a vaccine. Who all got Covishield here? Who all? Only one. 3 of you did not get any vaccine. Okay. Covishield is made up of chimpanzee gene. So you have chimpanzee gene in your body now. Okay. So Covishield uses a not the exact gene, it uses chimpanzee viral vector. Chimpanzee gene is used as a transporting agent, right? So what they use see what is the difference between Covaxin and Covishield? They are going to use a weakened strain of adenovirus. Adenovirus, what is adenovirus? It is a virus of common cold. So that is Shita. So that virus is used and it is transported using chimpanzee gene, right? And given as a injection, right? So, and to that they add this spike proteins. What is spike protein? So if there is a virus, there will be like this, right? On the COVID, this is spike protein. So spike proteins are added, not the entire virus is not added to the vaccine, just the spike protein is added. That is the mechanism, right? So spike protein is added, it is going to enhance the immunity system, immune system. Then mRNA vaccine, mRNA andre messenger RNA. So this is the simplest form of vaccine. What does it do andre? DNA na transcribe maadi, DNA will be changed into mRNA. That mRNA goes into the body and starts producing protein. That protein is going to fight the, so the antigen is also a form of protein. That protein is going to fight the coronavirus. That is the mRNA vaccine mechanism, right? So this mRNA vaccine, which is the vaccine you have, HGC019, that is a mRNA vaccine, HGC019. So what is the advantages of mRNA vaccine? They might ask why mRNA vaccine is good, messenger RNA vaccine. So one is it is going to directly produce the protein. So it need not get transcription like DNA. So direct protein and antigen protein will be produced, right? So there will be immediate immunity response. Immunity response will be immediate, right? So and also it can be manufactured very fast. mRNA vaccine manufacturing bega agute, then it is safe. It is non-infectious. Andre some other vaccines if you put, there is a probability of you getting COVID, but mRNA you won't get COVID, right? So then what else? Then it is fully synthetic. Fully synthetic and it is fully man-made, right? So it can be quickly manufactured, right? So it is a fully synthetic and do not require a host for growth. Andre it grows automatically. It does not require any vector to transport. That is the advantages of mRNA vaccine. So then we have CEPI mission, coalition, what is the full form of CEPI? This was in news during the COVID. CEPI. Coalition of Epidemic Preparedness Innovations, CEPI. So where is the headquarters? Norway. So we in India, we had IND-CEPI mission, IND-CEPI mission. What is IND-CEPI mission? India-centric epidemic preparedness through rapid vaccine development. So you just remember that there was a IND-CEPI mission, IND-CEPI mission. So it is a part of global CEPI mission. So what they do, that is, they do development of vaccines. They develop the vaccines, right? So who is implementing this IND-CEPI mission? It is implemented by VIRAT, biotechnology, whatever the authority I told, that authority is implementing the IND-CEPI mission. Next, I am going to talk about genetic testing. Genetic testing. So, what is a genetic testing? See, there are two types. One is genetic testing, examining the DNA. Examining the DNA is called as genetic testing. So, DNA can be examined for what? To find the spiral, what kind of base pairs are there. That is one thing. Second thing is to find out if the DNA is mutated, if there is any cancerous growth of cells. So because of that, this DNA genetic testing can be done. So there are two major types of gene test or genetic testing. What are the two major types, right? So one is say there will be DNA probes. So DNA will be there. So for the DNA there will be complementary DNA. So andre one is there will be DNA. For this DNA there will be another DNA. So what they do, this complementary DNA will be injected into a person, and this complementary DNA is going to get attached to this DNA. So once it get attached to the DNA, if the DNA is mutated, if it has changed its form, then it is going to flag it, and there is a change in this DNA, please notice anta, it is going to flag it, right? So that is the first type of testing, normal DNA and mutated DNA, complementary DNA. Complementary DNA goes and checks the normal DNA and finds out whether it is mutated, whether the gene is mutated or not. Then you have second type. Second type of this genetic testing is, there will be normal DNA irutte, then some of the disease DNA is there. So normal DNA and disease DNA will be checked. That is second type of testing. So there are two types of testing. What are the uses of this genetic testing? What are the uses? One is disease prevention, to prevent any disease, that is the first use. Then newborn screening, andre if the newborn is having any disease, that can be screened. Then what else? If the disease screening, andre if an adult disease screening, if an adult is having any disease or probability of getting disease in future, that can also be screened. And also forensic testing, this can be also used for forensic testing, genetic testing. So next I am going to talk about gene therapy. What do you mean by gene therapy? Use of genes to treat diseases. To treat diseases is called as gene therapy. Use of genetics. Use of genes to treat diseases or prevent the disease is called as gene therapy. So what they do, instead of medicine, they are going to insert a gene into a patient cell, and that gene is going to repair the diseased tissues. That is the gene therapy, right? So it is used for curing genetic diseases and also grave diseases like cancer, HIV and all those things. So what is the functioning mechanism? It is going to replace the defective genes. Whatever the defective genes are there, it is going to replace it. So what are the types of gene therapy? There are two types of gene therapy. One is somatic gene therapy and second is germline gene therapy. What is somatic gene therapy? So it is also called as a body gene therapy. Means what? It is going to target the genome of a person. Genome andre collection of genes. It is going to target the genes of a person and alter it to prevent the disease. That is the somatic gene therapy. So this somatic gene therapy, the genes are not going to get transferred from one generation to next generation. That is somatic gene therapy. Then you have germline gene therapy. What do you mean by germline gene therapy? Germline gene therapy andre, see here the changes inserted by, if you insert a gene there will be changes Allah. That change will get transferred from one generation to next generation. That is what germline gene therapy. So wow, how does the gene therapy can be implemented? There are two ways. One is ex vivo, another is in vivo. What do you mean by ex vivo? Ex vivo andre outside the body. If the gene therapy, if a person's gene is taken out, then the genetic alteration will be done and reinserted. Then that is ex vivo gene therapy. In vivo andre within the person body, if the gene therapy is carried out, that is in vivo gene therapy, right? So next I am going to talk about genetic diseases. See, these genetic diseases are also rare diseases, also called orphan diseases. Andre, it affects very small section of population. And why orphan diseases? Andre, there is not much medicine manufactured for these diseases. That is why they are called as orphan diseases, right? So this affects very small percentage of people, right? Especially the pediatric group. Pediatric andre, what is pediatrics? What is pediatrics? What is geriatrics? So you have never been to a pediatric. Okay, pediatric is a doctor. What doctor? Huh? You have never been to a pediatrician? Huh? Related to bone, that is orthopedics. You have never, pediatrician is a person who treats small kids. Children doctor are called as pediatrics. All of you have gone in your childhood. You have forgotten. Geriatrics related to old people. So treatment of old people is called as geriatrics. Pediatrics, small people. They are going to ask. This was asked in UPSC exam. Even I didn't know geriatrics. I messed up that answer. Okay. So approximately 70 million Indians or 7 crore Indians suffer from these rare diseases. What are the types of rare diseases? Hemophilia, thalassemia, sickle cell anemia, muscular dystrophy. All these are the forms of rare diseases. So this, with respect to these rare diseases, the Central Drug Standard Control Organization has brought in clinical trial rules. Andre clinical trials andre, the medicines will be undergoing clinical trials, right? So for that clinical trials, the rules are brought out. That is by the CDSCO, and so the drugs manufactured for treating these rare diseases are called as orphan drugs. Orphan drugs, right? Okay. So this is about genetic diseases. So next I am going to talk about what are the side effects of gene therapy. What are the concerns? One is, see, we are doing an experiment. We don't know how it is going to backfire. That person might die, right? So recently in America, they put a pig's heart into a person and the person died. within a month or something. Anyhow, he was going to die, he got a pig heart and died, right? So that is one thing. Then these genes can actually start functioning against the body. It can become immune to the body system, right? Starts manufacturing toxins. That is one thing. Then we don't know how these genes are going to function in the long term, right? That is also a shortcome. Then what else? This gene therapy involves multiple genes, not single gene. So if there is something going wrong, we don't know which gene malfunctioned. That is also one shortcoming. And gene therapy is highly costly, prohibitively costly. So these are the things. So with respect to gene therapy, Government of India, Indian Council of Medical Research has given a national guidelines on gene therapy. 2019, it has given a national guidelines on gene therapy. So what does it say? It says treatment of genetic disorders by gene therapy. That is what it says. So it says 70 million Indians or 7 crore Indians suffer from inherited genetic diseases. So there is a need for gene therapy, right? So this why there is a need for guidelines, Andre? One is the number of people who suffer. Second is these orphan diseases are ignored by pharmaceutical industry. They are not manufacturing the medicines because the number of patients are less. There is no profit for them. So they are not investing in the research and development, right? So that is one thing. And what else? So what is the objective of the guidelines? I told you, objective is to reduce the genetic disorders or bring genetically disordered people to cure the diseases of them. That is the objective, right? So what are the guidelines, specific guidelines? One is it uses the nucleic acid present in the nucleus for the therapeutic benefit, for therapy. Gene therapy, it is going to use the nucleic acid, and it says this gene therapy, the guideline says the gene therapy should be used scientifically and ethically. It should be used in a proper way, in a scientific and ethical manner. And this gene therapy, this will be monitored by a authority called as Gene Therapy Advisory and Evaluation Committee. This committee is going to monitor this, and also it focuses on training and infrastructure development for gene therapy. It is going to focus on training and infrastructure development. So what is the significance of this? Significance of these guidelines. One is the high burden. So high burden of diseases, we are going to reduce the burden, right? So what are the concerns? Concerns is cost. I told you, as I told, cost is a concern. So what can be done? One, government has to invest in research on gene therapy, then make it affordable and use the gene therapy ethically. Ethically, andre, don't put animal gene into humans and try to cure the disease. Use it ethically, right, in a proper fashion. And also use the gene therapy with the consent of the person. You should take the permission of the person before inserting the gene, right? So this gene therapy or genetics, genetic engineering is a huge industry. They say by the year 2024, it will grow into a $250 billion industry, and India has a huge potential to tap into this, right? So that is what they say. Next is gene doping. Gene doping. What is gene doping? What is doping? Where is it used? Doping, in which sector they use the doping? Sports. They use the doping in the sports. They are called performance enhancement drugs, right? So this is actually gene doping. They are going to use the genes, but not for therapeutic purpose, for non-therapeutic purpose. Mainly athletes to improve their performance. Actually, it has been prohibited by World Anti-Doping Agency, WADA, right? So what they do, what is the exact function mechanism andre, they are going to insert a gene. That gene is going to either increase certain functions or decrease certain functions. Andre, see, if there is a pain in the body, these genes are going to reduce the pain. Or if there is inflammation, UTA, and these genes are going to reduce that inflammation. All these things will be done by this gene, insertion of gene doping, right? So it is going to increase the performance, and unethically those persons are going to win the competitions, right, in the gene doping. So what are the uses? What do they do? It stimulates muscle growth increase agute. You know, right, certain bodybuilders, so they will be having so much body, but all of them are due to steroid body. They are not natural. Without steroids, it's impossible to build such bulky body. So they take steroids, which is very harmful to the body, right? So this gene doping is actually prohibited by World Anti-Doping Code given by WADA. World Anti-Doping Agency has given this code, right? So it gives an annual list of prohibited substances. Certain substances which are prohibited, it gives a list, right? So this word, there is an agency called World Anti-Doping Agency. In 2003, it actually bought in this code. This code was bought in 2013, World Anti-Doping Code in 2003 to ban the gene doping. To ban the gene doping, this was done, right? So that is in the year 2003, right? So this is about the gene doping. Then I'm going to talk about biotechnology in healthcare. How biotechnology is used in the healthcare, right? First is cloning. What do you mean by cloning? Cloning, see, there are two cells. Let's say this is one cell and this is one cell. Each cell, though, they are going to remove the nucleus. They are going to remove the nucleus and put the nucleus of this cell. That is cloning, right? Cloning of cells, right? Removal of nucleus from one cell and placing it in the unfertilized egg cell of a different whose nucleus has been removed. Another yadrudaru nucleus na tagdi alla haktare. So that is, so the first cloned animal was a sheep called Dolly, manufactured in the year 1997. They also planned to clone human beings, but by then it was banned. Actually, last year or last to last year, there was one person who had carried out human cloning, one scientist in China. He was banned and jailed, right? So all his licenses were taken away. So human cloning has been banned. So there are two types of cloning. One is reproductive cloning. Reproductive cloning andre, what do they do? So they are going to place the cell in the uterus of the human. So woman. So in the uterus, the that cell is going to grow into an embryo. That is reproductive cloning. That is cloning used for reproductive purposes is called as reproductive cloning. Then the other type of cloning is therapeutic cloning. Therapeutic cloning andre, cloning is done. That is, the nucleus is removed and put into some other cell. That mechanism is used for some therapy, treating of some disease. That is therapeutic cloning, right? So there are, what is the difference between therapeutic cloning and human reproductive cloning? Andre, see, therapeutic cloning, the cells are grown outside the body. So in a test tube that we call test tube baby, right? That kind of a thing. So in reproductive cloning, the cloning will be done within the body of the human being, right? So this therapeutic cloning is used to treat specific disease. So whereas this reproductive cloning is used for reproduction, test tube baby and all those things. So therapeutic cloning takes just few weeks. Reproductive cloning, how many months a baby takes? 9 months, right? So then here in therapeutic cloning you don't need surrogate mother. Surrogate mother andre badige tayandiru, right? Here you don't need surrogate mother, whereas in human reproductive cloning you need surrogate mother, right? Surrogate mother who will carry that cell. Then what are the ethical implications and medical implications? All those things not treated. Next I am going to talk about stem cell therapy. What is a stem cell therapy? So it is a regenerative medicine. Stem cell therapy is a regenerative medicine. Regenerative andre some part is damaged, that part has to be regenerated using stem cells. Stem cells, that is a regenerative medicine to repair the damaged tissue. To repair the damaged tissue, the stem cells are used, right? So it is used after organ transplantation. Organ transplantation ಮಾಡ್ತಾರಲ್ಲ, later on they use the stem cell therapy so that the organ gets adjusted to the new body, right? Okay. So stem cells, so they are actually, see, the stem cells are the raw materials of our body. So these stem cells can take the form of any other cell. It can become heart cell, it can become liver cell, kidney cell. All these cells it can become, right? So it can take any form. So these stem cells splits and become daughter cells. Stem cell will be there, it is going to split and take the form of daughter cells, right? So this only the stem cells can take the form of any other cell. So no other cell, normal cells cannot take the form of stem cells. So what is the significance of studying stem cells? Why stem cell studies is important? See, the first significance is to understand the diseases, how diseases going to happen, right? So, and also to replace the sick cells, the cells which are sick, that has to be replaced. That is done during the stem cells. Then using it for therapy, carrying it to the stem cell therapy can be done. So there are 3 types of stem cells, 3 classes of stem cells are there. One is totipotent cell. 3 stem cells, very, very important. 3 types will be asked. One is totipotent cell. So totipotent cell andre, totipotent means total. So the entire organism can be derived from this cell. So entire human being can be manufactured or reproduced from this cell. That is a totipotent cell. So it can convert itself into any type of cell, So, so this totipotent cell, can be used for fetal growth also, andre for baby growth, for creating new babies. Next, so this totipotent cell is a form of pluripotent cell. So the major form is pluripotent cell, under which the totipotent cell comes. So this pluripotent cell, this is a specialized type of cell. So this pluripotent cell can get itself converted into 3 types of cells. So I told you these stem cells can get converted into any types of cells. Pluripotent andre multiple potent. It can take the form of any body, any organ, right? So 3 major body tissues this pluripotent cells can take form. One is ectoderm, that is nervous system. It can take the form of nervous system and skin tissues. It can become mesoderm. Mesoderm andre it can become bone tissue, muscle tissue or blood tissue, and also it can become endoderm. Endoderm andre it can take the form of interior gut lining. Gut lining it can take the form, right? So if this pluripotent cell further becomes specialized, so next level of pluripotent cell is multipotent cell. Multipotent cell. So see, multipotent cell andre it cannot become all the types of cell. The types of cell it can get converted into is limited. Andre it can take only some specific forms, not all forms. So totipotent, that is the entire body, it can take the form of entire body. Pluripotent, 3 types of cells, 3 types, and multipotent, some certain specific type of cells it can become. So what are the, so where do we extract the stem cells from? If there is a human body, from which part of the body can we extract the stem cells? So embryonic stem cells, embryonic stem cells can be extracted from human embryos. Embryos and so that is actually where the baby forms. So the egg will be formed, the egg will get converted into embryo, the basic, the primitive stage of human baby in the body. So from that, if you take the stem cells, that is embryonic stem cells. Then from bone marrow or fat, from there also you can take the stem cells. Then adult stem cells can be taken from anywhere, right? So then perinatal stem cells can be taken from umbilical cord. What is umbilical cord? umbilical, umbilical cord. What does it mean? See, when a baby is born, its navel is connected to mother's navel, right? So its navel is connected to the mother. So from that pipe only the food will be going to the baby. That is called as umbilical cord. That is actually very important. There are a lot of research going on with respect to umbilical cord. So this is a new technology, umbilical cord technology, which I am going to discuss now. So that is cord blood banking. Cord blood banking andre umbilical cord. In this umbilical cord there will be blood. That blood is actually being saved. Why? So in the later, when the baby grows up, if the baby suffers some damage to the body, the person suffers some damage, some diseases, this umbilical cord blood can be used to treat the person, to make the disease cured. That is why this umbilical cord blood is being saved. So cord blood banking. What is cord blood banking? So recently they say, some years back, so a person, a 7-year girl was saved from aplastic anemia. Aplastic anemia, it is a form of cancer, right? So she was cured of that cancer because of this umbilical cord blood, right? So that is for India's first dual cord blood transplant. That is cord blood transport, for the first time it takes place in India, right? So why there is a need for cord blood banking? Why that umbilical cord blood should be saved, right? One is, so there is lot of cancers, lot of type of cancers that are growing. So to save persons from that type of cancer, this umbilical cord blood should be saved. And also there is lot of blood disorders like aplastic anemia, thalassemia, right? So to cure from those diseases, umbilical cord blood is there, right? So then see, what is the speciality of umbilical cord blood andre? That blood includes lot of pluripotent cells, pluripotent stem cells. So they can take the form of any part of the body. So it can become heart tissue also, it can become liver tissue, kidney tissue, anything. So that is why it is important. So it can repair the entire body, right? It can repair the entire body. Okay. So this umbilical cord blood, they are saved in cryogenic facility. What do you mean by cryogenic? Cryogenic technology andre enu? Very simple, yaar. So storing something. So cryogenic means sub-zero temperature. Any process, there is a cryogenic engine used by ISRO for launching rockets. That GSLV Mark III uses what? Cryogenic engine alva. So they are using this cryogenic technology andre storing something or operating something with below 0 degree Celsius is called as cryogenic technology. So umbilical cord should also be stored in cryogenic facilities, that is sub-zero temperature, right? Okay, so this I told you why umbilical cord blood is important, because it is pluripotent stem cell which is present. So normal pluripotent stem cell after the adult body, once we grow into adults, it is very difficult to find. But umbilical cord is full of this, right? So this umbilical cord blood is stored in cryogenic technology under liquid nitrogen. Liquid nitrogen. So this stem cell can cure 70 types of blood disorders, 70 types of cancers and blood diseases. So there are blood banking. This will be stored in blood banks, private blood banks or public blood banks, both, right? So what are the concerns with respect to umbilical cord technology? So one, it is expensive. It is very expensive. And ethical concerns. Ethical concerns andre one person's cord blood might be given to some other person without asking his or her permission. That is there. And also ICMR doesn't recommend this. Indian Council of Medical Research doesn't support this kind of cell banking, right? And also, no matter what you do, certain genetic disorders cannot be cured, right? That is there, right? So this is with respect to umbilical cord technology. Next, I am going to talk about the convalescent plasma therapy. When was it used? Where was it used? Convalescent plasma therapy, CPT. COVID. So what is the color of plasma? Where will be the plasma? Where will be the blood? So it is the what part of blood? This is a fluid part of blood. Pure fluid is plasma, right? So what will be the color of plasma? Pink? Yellow. It will be yellow color. So our blood is red, whereas plasma is yellow, pale yellow, right? So the blood color is given by some other cell to the blood, what gives red color to the blood? Hemoglobin. So plasma dali, of course, there will be no hemoglobin. That's why there is no red color. So what is this? What is the mechanism of convalescent plasma therapy? It means, see, a person would be recovered from the COVID. In that person, there will be a lot of antigens, which is against COVID, anti-COVID antigens. So after 2 weeks, after testing negative, that person's plasma will be taken and given to a COVID suffering person. That person will be, what will happen to that person? He will get cured very fast. That is the use, right? So, you know, this blood has got red blood cells, white blood cells, platelets, plasma. All these things are removed and only plasma is given, right? So this was not the first time when plasma was used. It was used in 2018 Ebola outbreak. Ebola. Where was it? Which country or which continent? Ebola? Africa, Senegal mostly. Then even during H1N1, influenza virus pandemic, H1N1, which disease? Bird flu, na? What is H1N1? Hini influenza virus. What is Hini? H1N1. Swine flu, yaar. Hini is swine flu. Swine flu is Andijora. So what is bird flu? They are going to ask what is bird flu, what is swine flu. These are the type of questions they want to put so that we get confused. Bird flu H5N1. Write it down. Bird flu H5N1. H1N1 is swine flu. Okay. So there this plasma therapy was used. So how does it function? Andre, the antibodies are going to kill the COVID coronavirus will be killed, right? So this white blood cells iruttalla, they go and get attached to the virus and kill it. So you can go on Google. So they have done a microscopic study where how the white blood cell is going to capture the bacteria and kill it, right? You can see it, a microscopic video is there, right? So then now let's talk about the working. So I told you the working, two weeks after the patient has recovered, it will be extracted. So the serum is what they separate. Serum andre plasma will be separated and given. So this plasma will be having IgG antibodies. There are multiple types of antibodies in COVID. One type is IgG. IgG antibodies irutte, they fight the COVID-19 virus, right? So this is a type of passive immunization. What is difference between passive and active immunization? Active andre directly taking the injection. Passive immunization is indirect. This plasma therapy is a indirect type of immunization, right? So this is going to be effective for 3 to 4 days. So I'm talking about antibodies. What is antibody? It is anti to some pathogen. Some foreign body enters our body. This acts as an antibody. Right? It is anti and against that. Right? So what is the protein that is secreted by the antibodies? It is T lymphocytes. T lymphocyte is the protein that is secreted by the antibodies. Okay. So what is the difference between plasma therapy and vaccination? Plasma therapy is passive immunization, whereas vaccination is active immunization. Plasma therapy is short term. Active immunization is long term. That is the difference. But see, plasma therapy, so it is used for critical cases, to treat the critical patients. So what are the risks associated with plasma therapy? So it might lead to spread of, if the proper checking is not done, it might lead to spread of hepatitis B, HIV, all these type of virus. And also it might cause allergy. A person's body might not accept another person's blood. And also it might lead to lung damage. All these things are the negatives, right? So what should be done? There should be more research on convalescent plasma therapy. There should be randomized clinical trials. Randomized clinical trials, clinical trial, you know, right? Trial of some medicine or something, right? That is clinical trial. Randomized andre, random people are given, right? Random, so there will be a group of 100%, out of which random, some 50 to 60 random people are given medicines. So others are not given this medicine. That is randomized control trials should be done. And also there should be protocol, proper protocol. Andre, not everyone should be given a CVT. Only the person body which is accept the CVT, they should be given the convalescent plasma therapy, right? Okay. So tomorrow I am going to talk about the bioinformatics. The last topic under biotechnology, bioinformatics, how biotechnology is used in the various knowledge related procedures. Knowledge andre DNA profiling, DNA coding, all those things I am going to talk about. So if you have any doubt, please put it in the comment section or reach us on our Telegram channel, VHS Education Forum. We will be happy to help. We'll meet in the next class. Until then, take care of your health. Jai Hind. Namaste.